Remember from our last lecture that based on their discovery of the structure of the double helix, Watson and Crick had proposed a mechanism by which DNA could be copied in such a way as to exactly replicate the order of nucleotides in the polymer through the complementary base pairing of those nucleotides with each other across the two strands of the double helix. Now, what they specifically suggested, you'll recall, is that somehow when the molecule is copied, replicated, the double helix must unzip, and then the two single strands would provide a template with, for the production of a complementary strand on the other side. And this idea they called the semiconservative model for DNA replication. We're going to begin today's lecture by asking how this model was tested, which is a very interesting story. And then we're going to go on and look in a little detail at the molecular mechanisms that we now know accomplish exactly this copying task. All right, let's remind ourselves of where we were. Remember that Watson and Crick's semi-conservative model contrasts with a couple of other possibilities for how DNA could possibly replicate that we mentioned last time. There's the conservative model, which suggests that both strands in the original DNA double helix stay together during replication. And there's the dispersive model, which suggests that both strands are not only separated, but even broken up into smaller pieces during replication. As I said last time, deciding which of these models was the correct one seems like it should be pretty easy to do because they make very different predictions. The prediction of Watson and Crick's semi-conservative hypothesis is that each of the two daughter double helices after one round of replication should be made up of one old strand entirely intact and one new strand also intact. The conservative model predicts that after replication, one of the double helices that result will be entirely older. Both strands will be older. The original parent, whereas the other helix, the copy or the daughter, will be entirely made up of newly synthesized DNA. And finally, the dispersive model predicts that both strands both of the daughter double helices will be made up of just a mixture of old and new synthesized DNA. Now, this sounds simple, but as I told you, the difficulty was figuring out how to actually test that, because we need some way to be able to determine what's old and what's new DNA after a replication. And it took several years before anybody figured out how to do this. This brings us to a pair of researchers named Matthew Meselson, who was a graduate student at Caltech at the time, and Franklin Stahl, a postdoc also at Caltech. In 1957, a few years after Watson and Crick's work, they came up with a novel method for distinguishing new and old DNA during replication. Let me explain how they did that. First, they grew bacteria in two different kinds of culture media. One of these culture media had normal nitrogen in it. Normal nitrogen is N14. And the other media had a heavier isotope of nitrogen in it, N15. Now, this isotope of nitrogen, N15, is not a radioactive isotope. It's just a little bit heavier, literally because it has one extra neutron associated with the nitrogen. Not much heavier, but just a little bit. So the difference between N14 and N15 nitrogen is a slight difference in how heavy or how dense the atom is. Now, the point of culturing bacteria in these two different media is that the nitrogen in those media would be taken up and incorporated into any new biological molecules that were being synthesized, and specifically would be taken up and incorporated into any new DNA that was being synthesized. Now, if you culture bacteria for some period of time, in other words, over what would be many, many generations of the bacteria, which must involve many, many replications of the DNA that they have, then you can assume after some period of time that all of the nitrogen that's incorporated in that DNA will either have N14 or N15, depending on the culture media in which you're growing it. In this way, Meselson and Stahl could essentially label old and new DNA by how heavy, or how dense really, that DNA was. Now, As I said, and as you can imagine, the density difference between DNA that had been made with N15 as compared to DNA that was made with N14 is a really, really small difference. So the real trick, and this was what was really the clever part, was figuring out how to very accurately essentially weigh or measure the densities of these kinds of DNA. To do this, Meselson and Stahl devised a new kind of procedure called density gradient centrifugation. And this density gradient centrifugation was a way that allowed them literally to sort out DNA according to how dense it was. Now, the idea behind this is actually similar to the reason why swimmers don't sink in the Great Salt Lake or the Red Sea. If the density of a liquid and an object in it are more or less the same, then the object will neither sink nor float. It'll just sort of stay where you put it. So if you add a lot of salt to water, actually it becomes the same density as, say, our own tissues, and so you don't sink in that water, you just sort of float. In fact, it might even be more dense, and so you end up having a hard time getting into it. Now, the interesting thing is that if you have a gradient of densities, in other words, you have some range of high to low densities in some liquid medium, then objects of slightly different densities will sort themselves out, and they'll end up along that gradient at exactly where their own density matches the density of that point in this density gradient. So that's the idea that Meselson and Stahl had. Now, how do you create a density gradient that you're going to use to measure DNA? Well, what they did was they after trying a number of different kinds of solutions, found a compound called cesium chloride. This is a salt of a heavy metal that, when it's put into solution, has approximately the same density as DNA. Again, this is like the Great Salt Lake and the swimmer. Now, further, what they then did was take a tube of cesium chloride solution and centrifuge it. Now remember, we mentioned this last time, if you centrifuge a tube, what happens is the heavier stuff, the more dense stuff, goes to the end of the tube and the lighter stuff stays at the top. Now what Meselson and Stahl had to do with this experiment was centrifuge the cesium chloride solution enormously quickly. They actually spun it around so fast that they created 100,000 g-forces, which is really, really fast. So this is called ultracentrifugation. And they did it for a number of days. So they would have these cesium chloride tubes spinning around really fast for a number of days, but at the end of that point, you would get a density gradient of the cesium chloride along the tube. Remember, cesium chloride is about the same density as DNA. If you spin it in the centrifuge fast enough and long enough, you get a gradient of densities that will then allow you to actually find exactly where along that gradient DNA falls. Or another way to put it is that if you then take some DNA and you put it in with that tube and you spin it around, the DNA will ordinarily just be dispersed in that tube because it's more or less the same density as the cesium chloride. But as the density gradient of cesium chloride develops, the DNA will all coalesce in a single band. And that band will be at a position along the length of the tube that you're spinning that corresponds exactly to the density of DNA at that point in the tube, matching the density of the cesium chloride solution. This method was so sensitive that Meselson and Stahl determined that they could tell the difference between N15 DNA and N14 DNA. In other words, the very slight density difference they got from adding one neutron to a nitrogen that got incorporated into the DNA would be sorted out along this density gradient. Okay, so that's the technique. Armed with this technique, Meselson and Stahl then did the following experiment, which should be sort of obvious based on what we've talked about in terms of the predictions that we have outlined, to test the semi-conservative model of DNA replication. They took a culture of N15 bacteria. Remember, this is a culture of bacteria that have been growing in this heavy nitrogen medium for a long time, where they could assume that all of the DNA in it was going to be have incorporated the heavier nitrogen 15. They then took some of those bacteria, N15 DNA bacteria, and transferred them to a culture flask that had N14. Now those bacteria, when they started to replicate their DNA so that they could divide, would start incorporating the lighter nitrogen. Any new DNA produced by those bacteria would be lighter than the old DNA that they had. They waited for about 20 minutes, which is long enough for just one round of DNA replication. In other words, the bacteria they used, would replicate themselves, would divide about once every 20 minutes. So if they took these heavy DNA bacteria and put them into a light DNA medium and waited 20 minutes, they would know that pretty much all of the bacteria in there would have divided once and therefore undergone just one round of DNA replication. Then they took the bacteria out and extracted the DNA from them and used this density centrifugation method I just described to determine what the densities of the DNA in the sample was. Now remember, the conservative model predicts that at this point there should be two separate sets of DNA. There should be lighter DNA and heavier DNA. The new DNA is all going to be lighter and the old DNA is all going to be heavier because the parent strand stays intact. So after one replication, you should have some DNA that's heavy and some DNA, the newly synthesized DNA, that's all light because they're in a double helix. This is not the result that they observed. In fact, what they saw instead of a light band and a heavy band was just one intermediate band. So they could rule out the conservative model directly. Now, they couldn't rule out the dispersive model. And the reason is after just one round of replication, both the dispersive and the semi-conservative models make the same prediction. In other words, each daughter double helix should be composed of half old DNA, that is half heavy DNA, and half new DNA, that is half light DNA. So all of the DNA in the sample after one replication should be at some intermediate weight, and that's what they saw. The dispersive hypothesis makes exactly the same prediction, and that's because all of the new and the old DNA just gets mixed up. So all of the DNA is going to be at some intermediate density. But if you wait for two replications, then all of a sudden you get a distinct difference in the predictions made by the semi-conservative and the dispersive models. After two replications, about 40 minutes, then you would expect for the semi-conservative model, you would still have an intermediate band, 50-50 new and old DNA, but now the new daughter DNA double helices that are produced should all be composed of lighter DNA. So you should have two bands, one in the middle and one that is higher up in the tube, which is lighter. The dispersive prediction would still predict that there would be only one band because all of the new and old DNA is just always mixed up, and therefore all of the DNA will be about the same density. The thing that should change is the position of that band along the gradient, because as more and more new DNA is incorporated, more and more light nitrogen is incorporated, that band should move higher and higher in the tube. And what Meselson and Stahl saw was, in fact, the creation now of two bands after two rounds of replication, which confirmed the prediction of the semi-conservative model of DNA replication. So it took some years, but Watson and Crick had it right, almost exactly right. Well, about 3 years after Watson and Crick's model was published, there was another really interesting discovery that was made by Arthur Kornberg, who's at Washington University in St. Louis. What he showed was that DNA can be replicated outside of a cell in a laboratory test tube. Now, at this point, Kornberg's interest was not so much in which model of replication was right, but instead was interested in specifically how replication occurred. Now, Watson and Crick, in their model, because of the complementary base pairing between the two strands, had actually suggested that the replication of DNA may not actually even require an enzyme. If you could just somehow unzip that molecule, they thought that the new molecules, the daughter molecules, might just self-assemble because the complementary base pairing would bring in all the appropriate nucleotides on the new complementary strands that were being made. Kornberg thought instead, though, that there must be some enzyme involved, and he set out to figure out what that enzyme was. Now, what Kornberg did was some old-fashioned, tedious, painstaking kind of work, because he said this enzyme, or he said there must be some enzyme, and so he started looking at all of the candidate proteins that are enzymes in bacteria cells and asking, do any of these synthesize DNA? So he would, I mean, this is simplifying it, of course, greatly, but he would essentially create culture medias where he would put three things into a test tube or a beaker. He would put some single-stranded DNA that should serve as a template for making new DNA, because we need a template. He would put in some of the nucleotides that the DNA had to be made of. These are the monomers. And then he'd take some candidate enzyme that he had purified from a bacterial cell and throw it in the mix. And he'd ask, do I get DNA synthesis? And he did this over and over and over, and with all sorts of candidate enzymes. But in fact, he did find one. He found an enzyme that he named DNA polymerase, which when it was put into the mix with a single strand of DNA that could serve as a template, with nucleotides that could serve as a monomer. These nucleotides actually were in a different form, and I'll mention that later. They actually were in a higher energy form that was needed to get this polymerization to occur. But he put in the nucleotide building blocks, and then he would put in this enzyme, which he called DNA polymerase, and he would get the synthesis of a new complementary strand on the old existing template strand that he added. This was really an important discovery, because this really opened up the modern era of molecular biology. The ability to find a particular enzyme that is the enzyme responsible for the biochemical task of actually making more DNA began what is now really almost 50 years of intense work to look at how that enzyme works, to look at the specific mechanisms by which DNA replication occurs. That's actually often how science works. There's an initial discovery, like Kornberg's discovery of DNA polymerase, and then generations of scientists come along and fill in the details. But it all started with Kornberg. And I don't have time in this course to tell you about all of the experiments, the painstaking and very clever work that went along the way. What I want to do now instead is to step through the mechanisms that we think are responsible for actually replicating DNA. Now, we know that it's replicated in a semiconservative fashion, and we know that it depends on complementary base pairing for that replication to occur accurately. But what actually happens when it's being replicated on a molecular level? So let's take a look at that. Well, the first thing that we know has to happen, as Watson and Crick predicted, is that the two sides of the DNA double strand have to be separated. And in fact, they are separated by an enzyme, not DNA polymerase in eukaryotic cells at least, but a different enzyme, an enzyme that's called helicase. By the way, you might notice a pattern here about how scientists name enzymes. They always end with an ace, and the name, except for some people who get a little fancy, usually tells you exactly what the enzyme does. DNA polymerase is the enzyme that polymerizes DNA. Helicase, it's a little bit harder there, but that's the enzyme that literally opens up the double helix. It breaks the hydrogen bonding between the two sides of the helix and starts spreading it out. Now, once helicase has done its job and would start spreading apart these two sides of the double helix, you can imagine that the complementary base pairing would then want to go and zip them back up again. So there's another set of proteins that come along right after that. These are generically called single-strand binding proteins. And these proteins come and sit on the now open DNA and hold it open. This is like putting a little chalk in there, a little block so that the thing can't zip back up. So they're really structurally stabilizing the single-stranded DNA, which would rather be a helix, at least rather on a molecular level. Now, I want to point out that the replication of a DNA double helix doesn't just start anywhere. Instead, it only starts in specific places on any one DNA molecule, and we call these places where it starts This is actually an obvious name, origins of replication. The point I want to make here is that these origins of replication themselves are not just arbitrary places on the DNA, but they're specific sequences of nucleotides on DNA that serve themselves as a special code, not for making proteins, but instead for starting the replication process. The way this works differs from those two kinds of cells we talked about before, prokaryotes, which are simpler cells, and eukaryotes, which are much more complex. In prokaryotes, the DNA exists as a single circle. It's actually a double helix that comes up and connects with its own other side. So it forms a continuous circle. It's a circular strand of DNA. That would be the chromosome for a bacteria, for a prokaryote. In eukaryotes, typically the DNA occurs not in circular strands, but just in linear strands. We call those chromosomes. And in fact, many eukaryotes will have several different linear strands, a number of different chromosomes. We have, for example, 46 chromosomes in our cells. Now, in prokaryotes, there's usually only a single place on that circular strand where DNA replication begins. But in eukaryotes, there may be many, hundreds or even thousands of special places, so that when those eukaryotic chromosomes, DNA strands, are being replicated, replication starts all over the place. And you can imagine that if you picture just a double strand of DNA and you start replication right in the middle here, then replication is going to be going out in two directions away from that origin of replication, forming what you might call a replication bubble. And if there are replication bubbles forming further down the line in either direction, eventually as those bubbles meet each other, they'll join up. Now, those details aren't really critical. What I want to do is to focus your attention at what's happening at that place where the DNA strand is opening up, which we call the replication fork, a Y-shaped kind of area, you can imagine, where the new strands of DNA are being made from the template of the now separate old strands of DNA, the template strands. This is where the major player in our process of replication, the enzyme that Kornberg found, DNA polymerase, does its work. Here's another important aside for you molecular biologists in the crowd. Since Kornberg's discovery of DNA polymerase, we found that there are actually a number of different kinds of DNA polymerase. Different kinds of DNA polymerase in prokaryotic cells versus eukaryotic cells, and even within the same kind of cell, different kinds of DNA polymerase that do slightly different parts of the job. But we're going to all call them, for the sake of this course, all DNA polymerase more generically. And what this enzyme does, as its name implies, is to simply polymerize the new nucleotides on the growing new strand of DNA as it's being complementarily matched to the older template strand. This polymerization requires energy. We're going to talk more about energy in detail on the molecular level in the third segment of the course. But let me mention now what I mentioned to you before in the lecture, and that is that the nucleotide monomers that are added on to the growing strand, the growing new strand, are in a slightly different form. They're in a form where instead of having one phosphate group on the nucleotide, they actually have 3 phosphates. And so we call them, and this is a big name, it's a deoxynucleoside triphosphate, or what we really call them are DNTPs for short. Those phosphates that are added provide a source of energy because bonds between adjacent phosphate groups are both high energy and very unstable, and so they're a useful way to get energy into a chemical reaction by a mechanism that we'll talk about in more detail again later on. Now, let me remind you that the bases that are being added on by DNA polymerase are being not chosen by DNA polymerase itself, but instead are being lined up through the complementary base pairing that's inherent in these nucleotides that are added, A with G, A with T, C with G, and so forth. All that DNA polymerase does is come to the next nucleotide that it sees there and say, okay, you're next, and build a chemical bond, a strong covalent bond, between the growing strand that it's working on and the next nucleotide that's going to be added in sequence. It just finds what's there, and what's there is there because of the complementary base pairing of that nucleotide with the corresponding nucleotide on the old template strand. I can't emphasize enough the significance of that idea because it's really the very thing that allows DNA to be replicated. And in a real sense, all else is just detail. Complementary base pairing. Okay, so far, this is all conceptually pretty simple. I mean, we have a few enzymes that open up the DNA strand, that stabilize it while it's open, and while it's held open by those single-stranded binding proteins, then DNA polymerase will come along and find whatever nucleotides have complementary base paired and add them to the strand. But there's an interesting problem that we've ignored. DNA polymerase is a remarkable molecule when you think about it. I mean, it does a really good job, and if it weren't for DNA polymerase, who knows where we would be today? Probably nowhere. But DNA polymerase has a couple of limitations. And one of the limitations that DNA polymerase has is that it only works in one direction. It only adds new monomers to the 3' end of a DNA strand. Remember 3' and 5'? The sugar of a nucleotide will have its main carbons numbered. The 3' end is attached normally in a nucleotide to a hydroxyl group, an OH, and the 5' end is what's attached to a phosphate group. And so when we're looking at the polarized strand of DNA, we're going from a sugar to a phosphate to a sugar, and what you would see is that the 5' end of one sugar connected to a phosphate is bonded to the 3' carbon on the next sugar in the chain. DNA polymerase actually works in the other direction. In other words, it needs to find an open 3' carbon with a hydroxyl group in OH to add a new nucleotide. So that's a limitation. Now, why does that matter? Well, it matters for an interesting reason. It matters because, if you'll recall, the two strands of DNA are antiparallel. That means that in a double helix, they're running in opposite directions relative to their 3' and 5' polarization. Here's a way to visualize this. Actually, you know, when you think of a double helix, it's a beautiful structure, but it's a little hard to keep it straight in your mind where 3' and 5' are sometimes. Imagine that you flattened a molecule of DNA, a double helix of DNA out, so it didn't spiral around. It was just flat. I like to call this the road-killed version of DNA. It would look more like a railroad track in this case instead of a spiral staircase, with the ties of the railroad track being the complementary base pairing of the nucleotides along the middle, and the rails being the sugar-phosphate backbones running along. We have on one of these rails the sugar-phosphate backbone going from 5' to 3', but in the other rail, it's going the other direction, from 3' to 5'. So why does that matter? DNA polymerase only works in one direction. That means that when the DNA double helix is opening up, only one of the strands at the replication fork is opening up in the right direction. In other words, the strand that's running, that is opening up in the 5' direction, will have its new strand, the newly synthesized strand, running antiparallel, being built in the 3' direction. And that's the direction that DNA polymerase can add on. So as the replication fork is opening up, DNA polymerase can just hop on and follow it down the railroad track. But the other side at this replication fork has to have something different going on, because it's opening up in the wrong direction. DNA polymerase has to keep jumping up to the opening of the replication fork and then synthesizing backwards, because that other side is going to have DNA polymerase running away from the direction of opening. We call these differences, by the way, the leading strand. That's the one where DNA polymerase can just follow it along, versus the lagging strand, where DNA polymerase has to make these short little jumps forward and backward. The discoverer, by the way, of the way that the lagging strand was synthesized was a Japanese scientist named Reiji Okazaki. And so we call these little pieces that are formed on the lagging strand Okazaki fragments. And what has to happen then is, once DNA polymerase has made a bunch of these fragments, then, as it turns out, another molecule has to come along and stitch them together. Actually, a few different molecules. Well, there are a number of other details that, of course, we could talk about in the way that DNA polymerase works, the way that DNA is replicated. But we've talked about the key issues here. And now, at the end, we see that not only did Watson and Crick have it right, but almost 50 years later, we've seen exactly how that mechanism works. And this is really quite important because our understanding of these mechanisms of DNA replication has been central to our understanding of how we ourselves can both look in detail at DNA by sequencing it, as we've sequenced the human genome, and in fact begin to manipulate that DNA as we start to change other organisms to our own whims. But we're not going to talk about that now. Next time, we're going to come back and we're going to take this process of DNA replication and put it into the broader context of how information flows from DNA out to where we really want it to go, which are the proteins that it makes.